molecules to these targeted areas and delivered them depending on the requirement of the delivery. So they can deliver the DNA, the protein molecules, as well as chemotherapeutic drugs. So firstly, this robot works on magnetoelectric phenomena. Like electromagnetism, it works on a bulk scale very well. Like all the motors, generators, dynamos, they work on electromagnetism phenomenon, and they have changed revolutionary in this modern world. Similarly, this magnetoelectricity, which is the converse of electromagnetism, have a very high potential at nanoscale as well as at microscale. So firstly, let me explain how magnetoelectricity work. A magnetoelectricity means by applying a magnetic field, we can generate electric field. Here, this is attained in a heterostructure where, for example, if we have two layers of magnetostrictive, that means you change the shape of the layer by magnetic field, you can generate an elastic wave. This elastic wave is passed on to the middle layer, which is a piezoelectric layer. Now this pressure wave is converted to electricity. That means with a AC magnetic field, we can generate an AC electric field, which is of the same modulation, same frequency as the AC magnetic field excitation. Okay. We have attained this magnetoelectric phenomenon in a nanostructure. So this is the structure where we have a core and a shell. The core is magnetostrictive cobalt ferrite and the shell is barium titanate. So when we apply a AC magnetic field, what happens is the core will start generating elastic waves. These elastic waves, once absorbed by the shell, generates electrical potential, very highly localized electrical potential, and at a amplitude of almost 15 millivolts at a scale of 100 nanometers, which is very high compared if we compare this in case of centimeters of electrodes being placed apart, and then we need to apply kilovolts of voltage through those electrodes to generate such a high electric field, localized electric field. The main hurdle was to fabricate such kind of structure individually because they have this layer of perovskite layer at the core, at the shell, and the spinel at the core. So they have a high lattice mismatch, and there was a requirement to get a single crystallinity because we want to keep the dipole, both electrical and magnetic dipoles, to be aligned in particular orientation so that they don't cancel out each other. Here is the nanostructure analysis where we have this core of cobalt ferrite in a single crystalline nature, as can be seen from electron diffraction. Then they have this shell over the core, which is also in single crystalline in nature. And here is one of the unique experiment of electron holography, where we can see this electromagnetic contribution difference between the core and the shell. As well as here, we can see how the cobalt and ferrite is all confined at the core of this structure, and the barium and titanate is all spread out throughout the surface of the structure. So we have this core shell structure, which will show the magnetoelectric mechanism, which I will show in the next slides. So this is one of the unique experiment again, which is called electron holography. Here we can see the invisible magnetic field lines as at the ambience of this core shell structure, and then we can see the electrical field lines being exposed or contained at the surface. Okay. Again, we can see this dipolar creation of positive and negative on the surface, which is exhibited by the electron wave. The electron wave of electron microscope have its magnetic and electric field. These magnetic field are creating this core shell structure to interact with each other to generate such kind of dipoles on the surface. Again, you can see that when we are using an AFM measurement, we can see the structure integrity, which always is in a same size as well as same structure, which is an elliptical structure. And here you can see what I was talking about, the North Pole and South Pole and the magnetic packet, which is around this structure. And this is a very unique measurement where you can see these invisible field lines, and these are the electrical hands of these robots. The switchability of this electrical field was confirmed by using a PFM measurement, piezo force microscopy, where we can see that we can we can change the electric field of the surface of these nanostructure by when analyzed with different tip voltages. And here is the MFM measurement, where we can see that at one side you have the deflection of the MFM tip, and another way you have the attraction, which shows the north pole and south pole creation, or a dipole, magnetic dipole creation on the structure. Okay. This is one of the measurement where we have analyzed that how the core and the shell is interacting with each other because of this elastic field, or the elastic wave being generated by the core, absorbed by the barium titanate shell, and there is a delay because of this absorption of this elastic wave and then transformation into electrical field. Okay. So the main hurdle was the heterostructure analysis, where you can see this, that whether the fabricated nanoparticles or these structures are high in integrity, and they don't lose this shell with exposure to alternating magnetic field particularly. So the size analysis, where we got this confirmation of the bulk scale nanostructures being of the same size of 120 nanometers. Now, there were two Nobel Prizes. One Nobel Prize in 1991 was about single ion channel in cells. And in 2003, again, the mechanistic study and the structural study of ion channels. So what is ion channel? In case of a cell with a membrane and the nucleus, what happens is, whenever there is an electromagnetic change in the vicinity of the cell membrane tries to open nanopore for the inflow outflow of sodium and potassium ion, and this will bring the equilibrium at the vicinity to the internal portion of the cell. Now, my main like hurdle, or you can say the main interest, was to engineer this activity of this nanopore opening by an electromagnetic device. Now, also this nanopore opening is very important. For example, if you touch a hot object, the cells nearby your hand which touches the hot heated part, they try to accumulate these thermal changes by inflow outflow of the sodium and potassium ion. But what happens is, when this is a very high heat shock, these generate a neuron which is passed on to the central nervous system or the brain, and then brain will say that remove your hands. So the first activity was to perform this kind of cellular mechanism by an electromagnetic device. Now, such kind of experiments were earlier also performed, where microelectrodes as well as electroporation cuvette were used to open the nanopores on the cell membrane, which also recovered themselves when the electric field was removed from the surface of the cell. But the problem is, this is highly uncontrolled way. You cannot have a single cell where you can perform this ion channel opening or electroporation on the cell. So to do that, uh, it is very important to achieve this fundamental, because you can transfer foreign molecules for the modification of the cell, particularly for T cell transfers for cancer treatment. Okay. Now, this nanoscale device of 120 nanometer can do this phenomena, which I will show you in my next slides, that their highly localized electrical field can perform this kind of electroporation on the surface of the cell. That means, using uh, this nanostructure, you can open one nanopore on this surface of a cell membrane. Okay. Now, why this nanopore is open in the on the cell membrane? So, cell membrane is basically made up of a phospholipid bilayer, which is a polar molecule. So, what happens is, whenever there is a change of electromagnetic energy near the cell, this polar molecule, this PO4 3 negative, which have 3 electron volts, in a cumulative way, they open the nanopores uh, because of the dislocation. The same thing which we are doing here is using this nanostructure, this nanorobot, to open the cell membrane by dislocating the phospholipid bilayer. And again, once this nanorobot passes inside, this will close itself. This will all close itself, because these phospholipid heads are connected with the fatty acid chain, and it finds the polar area and then again try to accumulate back to close this pore. So This phenomena of opening of the pore is as a excitation time, and the closing of the pore is the relaxation time. Now, this excitation time and relaxation time is different for normal cell, a healthy cell, as well as a cancer cell. Okay, we will use these differences to target a particular cancer cell, avoiding all the normal cells on the pathway. So let's see how it is done. Firstly, we try to perform this experiment in a cell where we can see that using a DC magnetic field, all the nanorobots are all attached to the cell membrane, the outside vicinity of the cell membrane. This is the same cell being picturized in a red filter, a green filter, and both of them combined together here. But in AC magnetic field, you can see that how these nano robots can pass inside these cells, creating these nanopores on the surface. Okay, and this was confirmed also again using the confocal microscopy, that how these particles are penetrating and are present inside the cell, inside vicinity of the cell. Now, to perform this experiment in a controlled environment, we used a microfluidic chamber, which have this small microfluidic chamber inside, which replicates the human vein conditions, both pressure and the velocity of a fluidic medium in this microfluidic chamber. And then the magnets were attached at the two ends, and this is all analyzed under a microscope, a fluorescent microscope with both bright field and colored image capability. As I said here, this was analyzed inside the microscope, the fluorescent microscope, and the magnets you can see here with the bipolar power supply. And you can see here that the cells were being arranged in this microfluidic chamber, and you can also see these robots which are tagged with the green dye to see their activity in an area, in a biocellular environment. Now here is the video, the first video, where I will show that how these nanorobots are able to create this electroporation in dynamically in cells. So this is the nanorobot where it is moving through this cell, which where the center cell try to goes up actually to create the lowest impedance pathway for these nanorobots. So this is a very quick process where it opens, penetrate, and then the pores will again close back. Now, this was for the electroporation, but then we said that we will use the relaxation time and excitation time to target the particular cancer cell. So this is the video where with another magnetic field, another modulated magnetic field, these nanorobots would avoid all the cells on the pathway. No matter what is the cellular density in the pathway, we will target one of the nanorobots here, which will to see its dynamic movement throughout the cells, and it will never interact with the cell, even if the density increases tenths of the fold. Okay. In the next part of this video also, I will show a slow motion of another nanorobot's interaction with the cell. So you can see that it never interacts with the cell in this pathway. Let's go to the next part. Here you can see that the real time, the time scale is also shown at the top. When these nanorobots are moving in an excitation of electromagnets in an AC magnetic field, they will always try to avoid the cells. Rather, they will gain the speed of their locomotion because of the electrostatic repulsion. Actually, when it faces a cell in the pathway, more cells, it will actually increase the speed. Why? Because we are generating a fluctuating negative field, and the cells also have the negative field because of the phosphate head. And because of this electrostatic repulsion and the modulated field, you can see it will pass on with a very high speed. And when it finds the particular targeted cell, it will just stop out there, and then you can switch off the magnetic field and perform the various experiments of either an electroporation or a transport mechanism. So using this experiments outcome, what we did was at one frequency we can penetrate inside the cell, at another frequency we can avoid the cells. Now in case of a normal cell and a cancer cell, what is the difference is basically in case of cancer cell, the Young's modulus of the because it increases its size from a healthy cell, it loses the Young's modulus of its cell membrane, and it have higher electrical energy. That means the excitation time and the relaxation time of opening of a pore and closing of a pore is of a cancer cell is different than a normal cell. So we have used this phenomenon, this experiment to perform experiment of the nanorobots with SCOPE3, which is carcinogenic ovarian cancer cell, and the no, healthy ovarian cells. And you can see that at one particular field modulated, they will enter inside the SCOPE3, but it will never enter inside the homic. Rather, they are avoiding it. So this was one of the phenomena of targeted cancer treatment, where the chemotherapy cannot differentiate between this normal cell and the cancer cells. Rather, they kill all of them, which have a very high side effect. So here is a more targeted form of treatment where we can deliver these drugs to these particular areas. Next phenomena, which we have observed of these nanorobot's interaction with the cell is the cellular transport. So now we are using a DC magnetic field because you saw that earlier in one DC field excitation, you can see that the nanorobots have trapped the cellular are attached to the cellular membrane are trapped there. So we are using now this fundamental to perform this experiment where in a DC magnetic field excitation, the nanorobots are attached to the cell themselves by this electrostatic bonding, and they start pushing the cells and covering this void areas and filling up in a very controlled way in these void areas, which is very unique because you can see that the pressure gradient is changing variably from this bigger region to the smaller region, but you can still push these cells to these areas and fill up these void areas in this microfluidic chamber, which resembles the human vein. Okay, so here maybe because of low resolution image, we cannot see the robots. So this is the next experiment where you can see that how the robots have been attached themselves with the electrostatic bonding on the cell membrane and then push these cells in a controlled way without disturbing the adhered cell areas. Yeah. So how we can use this? So in case of diseases like Alzheimer and Parkinson's, or even a transplanted organ, what happens is the neuronal connections breaks out. What is the reason of the neuronal connection break? Is because of the drying out of the cells, because of the water molecule being are released from these cells. So neuron doesn't find a conducting pathway. But recent experiments have shown that if fresh cells can be flowed through these minute areas, these minuscule areas, you can regenerate these neuronal pathways and again reconnect the central nervous system or the brain to those organs which were earlier disconnected with the brain. But there is no surgical method and no device which can perform this kind of cellular flow, particularly modified cells flow through these minuscule areas. So these nanorobots provide a unique technique where we can target the cells from an ambient nerve which has been dried and then push those cells to those dried areas to regenerate the neuronal pathway. Now, so using one DC magnetic field, the nanorobots can provide the treatment methods of diseases such as Alzheimer's, Parkinson, and reconstruction the neuronal pathways with transplanted organ. On the other hand, using the AC magnetic field modulated one, we can target the cancer cells, avoiding the healthy cells and deliver the chemotherapeutic drug on those cancer cells. So there will be almost no cytotoxicity. But one of the unique thing is that we are not keeping these nanorobots inside the cell, but rather after the treatment they are taken out, as I have shown you in my earlier video, that how they can penetrate from one cell to another cell without a disruption in the locomotion. Also, this technique can also be used for foreign molecule transfer as well as DNA transfer on targeted cells. So these are some of the biocompatibility tests which were needed to perform to prove that these robots are biocompatible. So the experiments were performed with human epithelial cell line as well as rat neuronal cell lines. These rat neuronal cell lines are very much active to electrical changes in the vicinity, and they shows a very high control biocompatibility. So next is, as I said, that these can also um, carry foreign molecules. So how they can carry the foreign molecules? First of all, on these rivers, on this nanostructure, we create this porous silica coating. On this porous silica coating, using magnetic pulses, we can generate whether the surface need to be negative or positive. Now, what happens is these biomolecules are particularly ions with either negative or positive charge. So, in this way, the chemotherapy drug are loaded on these pockets on the surface of the nanorobots by changing the surface potential. Also, in case of biomolecules, we have these tips where, for example, here you have the oxygen, so you have the two negative charge of the tip. Now, if we change the surface to a positive charge, what happens? These will stick to the surface as electrostatic bonding, and it can again be released by another magnetic pulse, which will change the surface potential to negative, and they will be released. So this mechanism was used in case of loading of anti-HIV drug, which is AGTTP. Here, using these bipolar magnetic pulses, we can load the long chain AGTTP molecule on the surface of these robots, as well as we can release these AGTTP again to particular sites or targeted sites by a magnetic field of different amplitude or a reverse amplitude. So at last, the same phenomena also can be used to load the DNA molecules in these pockets, and these can be delivered in those targeted sites by electroporating the cells and go inside the cells and deliver them. Okay. So in a cyst, the whole activity is this: we have core shell drug loaded on the top, and then we have these cancer cells and normal cells, and these can track these cancer cells, electroporate them, as well as they can deliver the drug by again a magnetic pulse. So this research was recognized worldwide through various articles, scientific journals, as well as the Guinness World Record, and also by National Science Foundation. And lastly, we cannot really inject these nanorobots in any particular area. Rather, they should be controlled, uh, controllably released in particular areas to perform these activities. So we have aligned these nanorobots in silica nanofiber and then made the capsules where we have this pH sensitive resin, and depending on the site, so this whole capsule after the injection will be carried out to particular site, and there the pH will dissolve this resin, and the robots are free now to perform the activity of cancer cell detection or neuronal cellular transport by a magnetic field excitation. Uh, so in my last slide here, this whole phenomena can be monitored using an magnetic resonance imaging. But on the other hand, also we can control this movement of the microcapsule using this octamag, where one of the experiment from ETH week have shown that how these robots can be controllably moved in a rabbit's eye at microscale precision. So lastly, I will acknowledge the funding agencies of Department of Defense from USA, National Science Foundation and National Science Foundation INM for sponsoring this research. And these are my references lastly. And thank you. And I'm open for the questions now.
Thank you, Shruti. So the floor is open for questions now. So maybe I have one question. Basically, what is the safe range? Because we are working with these magnetic and electrical fields. So is there any safe range? Because it has to be, planted inside a human body, right?
Yes, absolutely. But this is the trick. So since magnetic field at this low amplitude of 50 oersteds doesn't interact with any of the biological component. So what happens, no matter this, even if you put this very close to the outside of the body, they will not like have any toxicity, or you can say any deformation on the biological surfaces of our body. Secondly, the nanorobots are generating a very localized and very small electric field. So they can only uh, electroporate a particular cell when they are at micron scale distances. So there is really no hurdle of keeping them at a safe distance, because both the magnetic field and the electric field are not interacting with the ambient environment, rather to a particular target only.
Okay, so I had another question that how this robot is differentiating between normal cells and cancer cells.
Yeah, that's what I told in my earlier slides, right? I will go back to the slide. So
Yeah.
the differentiation is happening because of this change. So in case of a normal cell, we have this nanopore opening excitation time, which is by the electrostatic repulsion and phospholipid phosphate heads dislocation, right? Then when it goes back, they will again close these nanopores, which is the relaxation time because of the hydrophobic effect, where the fatty acid chain will try to accumulate back themselves. For example, like if you put drops of fat or on water surface, they will try to accumulate together because of hydrophobic effect. Okay. Now this mechanism of dislocation, this charge of dislocation is different for normal cell and cancer cell. So what happens is, if you create a pulse by these nanorobots, a electrical pulse, which is longer than the relaxation time of a cancer cell, but smaller than that of the normal cell. So what will happen is, they will avoid these normal cells and just particularly open the nanopore on the cell membrane of a cancer cell. That's how it is differentiating. But the best outcome here is they can do that while in a dynamic condition. They need not to be like standing at one particular area. Rather, they can just pass from one cell to another one by one.
Okay, thank you. Any other questions?
Hello.
Hello, Professor. Good
Yeah.
evening.
Yeah. Good evening. Good evening.
My name is Karan Yadav. I'm from MSc Cognitive Science here at IIT Delhi.
Yeah.
So, on the same line, I had a question about like the material it was used in a building the robot, and basically my concern was about safety. Like, what material was used to build the robot, and
Yes.
how it was built.
Yeah, I have explained it in my earlier slide that the material, the core was cobalt ferrite, the shell was barium titanate. I will go back to the slide. So you can see it is a cobalt ferrite and a barium titanate, and then it have another coating of silica on the top. So this was tested for the biocompatibility on different cell lines, among which one of the cell line is the human epithelial cell line, and the second is rat neuronal cell line, where we saw no toxicity at a 50 microgram per milliliter. So also one thing which I want to tell you, that each of the capsules which will be used with these nanorobots are in such a low quantity that they are much less than the dust we inhale every minute. So the amount of these nanorobots are used in a very minimal quantity for this treatment method. Yeah.
Okay, thank you, Professor.
Yeah, any other questions?
Hello. Good evening, sir.
Good evening.
So I wanted to ask that after the process, after the drug delivery, the particles will stay there only on the target site.
No, no, they are releasing out. They are coming out because that's what I showed in the video. So everything in our body is made up of cells, right? Now these nanorobots can penetrate inside each of the cell depending on the magnetic field excitation, right? So here you can see that after the release of the drug, they can pass on from one cell to another without being staying at that area. that is the unique thing in this research, that we are not keeping the robots inside the body after the treatment. Rather, they are just going out, and then they will be coming out of the skin surface by the sweat medium, rather than the urinary system, where they can create these stones because of,
Yes.
yeah, yeah. So they are using a very different method, and singularly they are coming out, and by the sweat itself on the pore of your body, they can just come out.
Okay, okay, okay. Okay, sir. Thank you, sir.
So, do we have further questions from our audience?
Yes, sir. I want to ask that can we treat some of the mental condition by using these kinds of nanorobots in our brain?
For example, I can give you one example, like I said about Alzheimer's. So what happened is, our brain have the highest connection with our memory. So whenever you have smell a perfume, for example, at a very small age, next time you will smell it, you will remember it, right? Now these memory veins, these neuronal connection of your memory to your brain, that often get dislocated or dried out because of hypertensions in old age people. Now that was one of the unique, that if you can freshly flow cells through these veins, you can again regenerate those neuronal pathways. That means your connection to your sensory organ, or your sense, or your memory location. This can be done by these nanorobots. One of the another thing is about this treatment method of the Parkinson disease, as well as reconnection of the transplanted organ, which often body assume it to be a foreign object and try to just disconnect with that, or doesn't have a connection of the brain to that. But then if using these nanorobots, we can improve this transplanted organs intake by the body.
Okay, sir. Thank you.
Yeah, so Zaya has asked that, sir, what are your thoughts on using this for cognitive enhancement? Are there any future prospects?
In case of cognitive enhancement, I can tell you that there are various features which need to be considered. For example, cognitive is relative to both intelligence as well as your sensory part. Someone have a very good reflex action. Someone might have a low reflex action, and then it will he will have more intelligence maybe. So this can be differentiated, but then enhancing them particularly needs a very big machine, rather, or a large number of nanorobots. So in that perspective, I really believe that these nanorobots will not be very influential subject of cognitive enhancement.
Okay, so Jaya, does that answer your question? Hopefully. Okay. So, if there are no further questions, thank you, Professor Shautik, for a really informative presentation. I just had the final point that has this been tested on some patients,
Right.
especially say the Alzheimer's patients?
This is one of the reason I'm working on the medical aspect with medical personals. So right now it is still in the animal testing phase, but I really believe that with this capsulated way and further in vivo studies, very soon it will come to human subjects, where we can use this on urging human subject, and the ultimate aim is to really eradicate the use of chemotherapy for at least first two stages of cancer, where we don't have a very large surface of the body to be exposed to radiation, rather more targeted form of treatment.
Okay, so it's really very interesting. Thank you again, Professor. So we would like to felicitate you with a token of appreciation from IHFC. So Saurav, if you can share the screen. Yeah, so this is a 150 euros Springer coupon So thank you again for providing a really nice talk on our platform. So we hope to listen to you more in future.
Thank you very much, and this is quite a respect for me. And I really believe that the organization of cobotics will really bring changes for the betterment of multiple fields, particularly because a lot of researchers from both healthcare as well as artificial intelligence and robotics, they have come together to share this platform and to bring multidisciplinary research to a common field.
Yeah, thank you again. So if audience have further questions, they can perhaps email Professor Soutik. Professor Soutik, you can share your email ID if it is okay
Yes,
for you.
I will write it right here.
Okay. And I'll be sharing the details on how you can access this coupon over emails. Okay.
Okay, sure.
Yeah. Thank you. Thank you everyone for joining in. Uh, we'll meet again on the next month. Thank you.
Have a nice day.